Welcome back everybody to daily lectures by New Age Cosmetic White EdTech Healthcare Consultancy. I'm Dr. Bobby, and we're on the subject of molecular biotechnology. We're on the specific subject of molecular diagnostics, and we're dealing with the real-time PCR. By labeling the DNA that is amplified in a PCR with a fluorescent dye and monitoring the fluorescence that results when the dye bound to double-stranded DNA is irradiated with the light of a certain wavelength, it is possible to watch the production of PCR products. Moreover, this approach allows one to quantify the amount of a specific DNA fragment in the starting material. Labeling the DNA is achieved by using any one of a variety of protocols. In the simplest approach to labeling DNA, the researchers add dyes that bind double-stranded DNA and emit fluorescence, and the fluorescence intensity increases in proportion to the concentration of double-stranded DNA. Real-time PCR may be described as occurring in 4 phases. In the first, or linear phase, which generally takes about 10 or 15 cycles, the fluorescence emission at each cycle has not yet been above the background level. In the early exponential phase, which is the phase 2, the amount of fluorescence reaches a threshold of which it is significantly higher than the background. The cycle of which this occurs is known as a threshold cycle. Depending upon the manufacture of the equipment, the CR value is inversely correlated with the amount of target DNA in the origin sample. And the amount doubles in each cycle under ideal conditions, where in the plateau stage, phase 4, the reaction components become limited and the measurements of the fluorescent intensity are no longer useful. To quantitate the amount of target DNA in a sample, a standard curve is generated by serially diluting a sample with a known number of copies of the target DNA and assuming all samples amplified with equal efficiency. To quantitate the amount of target DNA in a sample, the standard curve is first generated by serially diluting a sample with a known number of copies of the target DNA and assuming all samples amplified with equal efficiency. The CD values for each dilution are plotted against the starting amount of sample. The number of copies of a target DNA in a sample can be determined by obtaining the CD value for the sample and extrapolating the starting amount from the standard curve. In addition, since during the exponential phase the DNA doubles with each cycle, a sample that has 4 times the number of starting copies of the target sequence compared to another sample would require 2 fewer cycles of amplification to generate the same number of product standards. Among its many other uses, the real-time PCR has been used to monitor Cryptosporidium parvum, a waterborne protozoan parasite that is a causative agent of a range of human diseases, including persistent diarrhea and severe infections in infected individuals. This approach is likely to replace the more imprecise and time-consuming traditional methods of monitoring C. parvum infections, such as histological staining, Similarly, other researchers have reported using real-time PCR to quantitate S. enterica contamination in food samples. Using real PCTEM, quantitative S. enterica contaminated food samples. In this case, food samples like chicken and mung beans were tested, were rinsed with 100 to 250 ml of water or with a physiological saline solution. The liquid was filtered to remove particulate matter and then refiltered to capture the Salmonella sp cells. The cells were removed from the filter membrane, lysed and subjected to real-time PCR. In this case, the entire procedure took only 3 hours and was able to detect and quantitate cell numbers as low as 7 × 10⁻², colony-forming units, that is cells, per 100 ml of liquid, compared to the existing methodology. Real-time PCR offers a dramatic improvement in both the sensitivity of detection and the time that it takes to complete the analysis. This is a diagram which in addition to its use in the measurement of pathogenic agents in the environment, a variety of real-time PCR may be used to quantify the levels of a variety of mRNAs in different eukaryotic tissues or prokaryotic cells. In this case, since the initial target is RNA and not DNA, a reverse transcription RD step is needed before the real-time PCR. Now on to real-time PCR. By labeling the DNA that is amplified in a PCR with a fluorescent dye and monitoring the fluorescence that results when the dye bound to double-stranded DNA is irradiated with light of a certain wavelength, it is possible to watch the production of PCR products. Moreover, this approach allows one to quantify the amount of a specific DNA fragment in the starting material. Labeling the DNA is achieved using any one of a variety of protocols. The simplest approach to labeling DNA, researchers add dyes that bind to double-stranded DNA and emit fluorescence, and the fluorescence intensity increases in proportion to the concentration of double-stranded DNA. Real-time PCR may be described as occurring in 4 phases. In the first, or linear phase, which generally takes about 10 to 15 cycles, fluorescence emission at each cycle has not yet risen above the background level. In early exponential phase, the amount of fluorescence reaches a threshold at which it is significantly higher than the background. The cycle at which this occurs is known as the threshold cycle. The CD value is inversely correlated with the amount of target DNA in the original sample. During the exponential phase, the amount of product doubles in each cycle under ideal conditions, while in the plateau stage, the reaction components become limited and measurements of the fluorescence intensity are no longer useful. To quantitate the amount of target DNA in a sample, a standard curve is first generated by serially diluting a sample with a known number of copies of the target DNA and assuming all samples are amplified with equal efficiency. The CD values for each dilution are plotted against the starting amount of sample. The number of copies of a target DNA in a sample can be determined by obtaining the CD value for the sample and extrapolating the starting amount from the standard curve. In addition, since during the exponential phase the DNA doubles with each cycle, a sample that has 4 times the number of starting copies of the target sequence compared to another sample would require 2 fewer cycles of amplification to generate the same number of product strands. strands. Among its many other uses, the real-time PCR has been used to monitor Cytosporidium parvum, a waterborne protozoan parasite that is the causative agent of a range of human diseases, including persistent diarrhea and severe infections in infected individuals. This approach is likely to replace the more imprecise and time-consuming traditional methods of monitoring C. parvum infections, such as histological staining. Similarly, other researchers have reported using real-time PCR to quantitate S. enterica contamination in food samples. In this case, food samples, chicken and mung beans were tested, were rinsed with 100 to 250 ml of water or with a physiologically saline solution. The liquid was filtered to remove particulate matter and then filtered to capture the Salmonella sp cells. The cells were removed from the filter membrane, lysed and subjected to a real-time PCR. In this case, the entire procedure took only approximately 3 hours and was able to detect the quantitate cell numbers as low as 7 by 10 raised to 2 colony forming units, that is cells per 100 ml of liquid. Compared to the existing methodology, real-time PCR offers a dramatic improvement in both the sensitivity of detection and the time that it takes to complete the analysis. In addition to its use in the measurement of pathogenic agents in the environment, a variant of real-time PCR may be used to quantitate the levels of a variety of mRNAs, in particular in different eukaryotic tissues of prokaryotic cells. In this case, since the initial target is RNA and not DNA, a robust transcription RT step is needed before the real-time PCR. In the first step of real-time RT-PCR, the mRNA sample is reverse transcribed to generate complementary DNA, cDNA. This may be done in the same tube as a subsequent PCR, or the RT reaction and PCR may be carried out in separate tubes. Many of the more traditional methods of monitoring gene expression including northern hybridization, ribonuclease RNAs, protection assays, and RNA dot blot hybridizations, are both limited in sensitivity and difficult to quantify. However, with the increasingly popular technique of real-time RT-PCR, it is possible to detect and quantify mRNA levels that are about 10,000 to 100,000 fold lower than those measurable by traditional techniques. With real-time PCR, with real-time RT-PCR, even a single copy of the transcript may be detected. The very low levels of RNA that are required for this procedure make it the method of choice for monitoring mRNA levels. The next topic will be immunoquantitative real-time PCR. The detection limits of many commercially available immunological methods for measuring levels of pathogenic microorganisms are sufficient to perceive low but still potentially dangerous levels of these organisms. Notwithstanding the high degree of specificity that antibodies provide, it will be advantageous to be able to increase the sensitivity of various immunological assay procedures. One way to do this is to develop a protocol that combines the specificity of antibodies with the sensitivity of PCR. So, in relation to this, the figure on 9.23 compares an ELISA-type protocol with an immunoquantitative real-time PCR procedure. With the ELISA method, figure 9.23a, the first antibody is coupled to the surface of a microtiter plate, the added antigen binds to the first antibody. When the second antibody is added, it binds to a different epitope on the antigen. The bound antigen is visualized by the action of alkaline phosphatase bound to the second antibody, which turns a colorless substrate into a colored product. With the immunoquantitative real-time PCR procedure, instead of alkaline phosphatase, a streptobutene biotin complex links the second antibody to a 246BP DNA fragment with a known sequence. Once the immunological complex is formed, it may be visualized and quantified by performing real-time PCR in the well of the microtiter plate, thereby significantly amplifying the signal of the immunological complex. In fact, it has been estimated that the procedure is approximately 1,000-fold more sensitive than ELISA. Ancestry determination. By examining a number of different single nucleotide polymorphisms, SNPs, that is minor variations of DNA sequence, in an individual and comparing the pattern of the SNPs to those of other individuals in the population, it is possible to infer information regarding an individual's ancestry. For an analysis of an individual's ancestry, 3 different types of DNA can be examined. Autosomal DNA, which includes all of a person's DNA except for the X and Y chromosomes, and mitochondrial DNA, which originates from a combination of a person's parents' DNA, paternal DNA, that is the Y chromosome, which is passed on from father to son, and maternal DNA, that is mitochondrial DNA and X chromosome, which is passed on from a mother to all of her children. To perform an analysis of an individual's ancestry or for paternity testing or for forensic analysis, DNA is typically extracted and purified from buccal swabs, that is from cheek cells, or from blood samples. Stretches of DNA are then amplified by PCR using primers that target specific regions of the genome. The DNA samples are then separated by size on a small column by a technique known as capillary electrophoresis. This method has generally replaced gel electrophoresis, which was previously used to separate these small DNA fragments, but was slower and less amenable to automation. If the PCR primers are labeled with fluorescent dyes before the PCR amplification reaction, fluorescent samples are eluted from the capillary column in a characteristic pattern of bands, in much the same way that DNA bands form a pattern unique to a particular fragment following gel electrophoresis. The sizes of the amplified DNA bands in specific regions are determined and are referred to as alleles. With autosomal DNA, each person should have 2 alleles at each side, one from the father and one from the mother. Each known allele has a determined frequency of occurrence in the general population among various ethnic groups. After testing for approximately 150 to 350 different SNPs, the frequency of certain alleles in an individual is compared to the frequency of those alleles in various ethnic groups. This provides an indication of the ethnic background or ancestry of that individual. For example, this type of testing may indicate that an individual is genetically 70% Northern European and 17% Middle Eastern and 13% Native American. Since mitochondrial DNA changes only very little over many generations, characterization of mitochondrial DNA is an ideal means of tracking migration over many hundreds of generations of human genetic history. Analysis of mitochondrial DNA indicates that the observed genetic variations in human populations may be divided into divisions of ethnically similar individuals called haplotypes. Table 9.3 summarizes the currently accepted mitochondrial DNA haplotypes and the groups associated with these haplotypes. The root of all human lineages is the L groups in Africa. All other groups diverged from these groups after early human beings began to migrate out of Africa around 150,000 years ago. Analysis of parental Y chromosome haplotypes has been used to examine the claim that all members of the priestly line of Jewish males called Kohanim are descended from the family of the biblical Aaron, the brother of Moses. According to Jewish tradition, membership in this priestly line may be acquired only by males whose biological fathers are Kohanim. When Y-linked genetic markers were examined among Jews claiming to be Kohanim, as well as men who either were not Jewish or did not claim to be Kohanim, the limited variation in the markers examined was entirely consistent with a 3,300-year-old origin of this priestly line in a single male or a male group of related males. Interestingly, these same Y chromosome genetic markers are found among the Lemba, a South African tribal group claiming paternal kinship with the Jews of Yemen. By mid-2008, there were nearly 30 companies marketing genetic ancestry kits. Each test costs from about $100 to $900. Moreover, in the few years that they have been available, more than 500,000 people have purchased these tests, and there is every indication that the demand will continue to grow. Some geneticists, anthropologists, and epidemiologists have publicly expressed concern that naive laypersons anxious for definitive information about their personal genetic ancestry often misinterpret the results of these tests. For example, the fact that a particular allele or haplotype is quite common in certain populations does not mean that its presence is a definite diagnostic of membership in those populations. There is often high generic diversity within populations, and gene flow can readily occur between populations. Thus, a particular allele could have been inherited from a population in which it is less common rather than from the population in which it appears to be diagnostic, leading some individuals to mistakenly believe that they are genetically part of a particular ethnic or racial group. Animal species determination. In many parts of the world, numerous large mammalian species have been hunted to the verge of extinction because of the trade in their skin, bones, horns, or other body parts. As part of an effort to stem the illegal traffic in wild animal remains and enforce international conventions designed to prevent this trade, a number of laboratories have been set up to determine the species of organisms origin from powdered animal remains so prepared because of their supposed medicine properties. Currently, the method of choice for animal species determination involves genotyping. More specifically, using PCR, a portion of the animal cytochrome B gene, which is found in the mitochondrial DNA, is amplified and then sequenced. This locus was chosen because it is both sufficiently conserved so that it is present in all mammals and sufficiently polymorphic that members of different but closely related species can be distinguished. The primers used in this method amplify a 486pp DNA fragment, which is sufficient for DNA sequencing and to identify most mammals despite the fact that the DNA sample is often somewhat degraded. To ensure that the high quality data are obtained, it is necessary to start with 20 to 50 ng of DNA template, and 2, use a control DNA sample, usually mouse or cow DNA that is treated in parallel to the target DNA. Automated DNA analysis. More than 1,400 different organisms, bacteria, fungi, virus, and protozoa have been identified as being pathogenic to humans. Moreover, new pathogens are constantly being identified and characterized. To limit the damage from either a natural epidemic or a bioterrorist attack, it is necessary to rapidly identify the organisms that is a source of infectious outbreaks so that appropriate public health measures may be instituted as rapidly as possible. In addition to human diseases, it is also important to rapidly identify the causative agent of outbreaks of animal and plant diseases. By performing a combination of PCR and electrospray ionization mass spectrometry, it is possible to rapidly and accurately identify a wide range of human pathogens. Small aliquots of the DNA that is isolated from a test sample are placed into a number of different wells of a microtiter plate. Each well contains a pair of PCR primers that have been designed to amplify a gene product from a broad group of organisms within a selected domain of microbial life. For example, workers have reported using 12 primers that are targeted to universally conserved sequences and 6 primers that are targeted to broad divisions of microbial life, such as bacilli. The products of each of the PCRs are electrosprayed into a mass spectrometer. The DNA-based sequences of the various samples are determined. This technique allows scientists to very rapidly hone in on the nature of an infectious agent, of course. This technique is facilitated by the fact that the genomic DNA sequences of a large number of microbes have already been determined. Moreover, with the very rapid progress that has been made recently in DNA sequence analysis, it is reasonable to expect that all known pathogens will be sequenced fully by 2015. Molecular diagnosis of genetic diseases. The ability to diagnose the occurrence of specific inherited diseases in humans at the genetic level makes it possible for individuals to discover whether their offspring are at risk. DNA analysis can be used for the identification of carriers of hereditary disorders, for prenatal diagnosis of serious genetic conditions, and for early diagnosis before the onset of symptoms. Tests at the DNA level are definitive for determining the existence of specific genetic mutations. Previously, genetic testing relied almost exclusively on biochemical assays that scored either the presence or the absence of a gene product. A DNA-based test does not, however, require expression for detection of the mutant gene, thereby making it theoretically possible to develop screening assays for all single-gene diseases. Screening for cystic fibrosis. Often, screening for genetic diseases can be rather complex. This reflects the fact that instead of a disease being the consequence of a single bacteria, let me repeat that sentence for you. This reflects the fact that instead of a disease being consequence of a single alteration to the wild-type DNA, as is the case with sickle cell anemia, many diseases are caused by any one of a large number of genetic alterations to normal DNA for that gene. For example, cystic fibrosis, one of the most common lethal autosomal recessive disorders in Europeans and their descendants, with an increase of approximately 1 in every 2,500 live births and a carrier frequency of approximately 1 in 29, is caused by mutations to the cystic fibrosis transmembrane conductance regulator CFTR gene that results in defects in chloride ion transport. That there are currently nearly 1,400 known mutations to the CFTR gene that can result in the development of cystic fibrosis. Screening individuals who may be at risk for cystic fibrosis for 1,400 different mutations is a daunting task. Fortunately, some of the mutations that cause cystic fibrosis are much more common than others. In fact, over 90% of cystic fibrosis patients carry at least one delta F508 allele, and nearly 50% of cystic fibrosis cases are individuals who are homozygous for delta F508. Despite the fact that separate tests are required for each mutation, it is estimated that screening individuals for delta F508 and for the next 20 most common mutations should identify approximately 98% of cystic fibrosis affected individuals and carriers. Current diagnostic tests for cystic fibrosis includes several different techniques. Right? One of the most widely used methods is allele-specific oligonucleotide dot plots, also called allele-specific hybridization. With this technique, genomic DNA or cDNA from an individual is amplified by PCR, and following transfer to a member is hybridized separately to label oligonucleotide probes for the mutant, usually a delta F508 and wild-type genes. In this way, it is possible to distinguish between normal individuals, cystic fibrosis carriers, and cystic fibrosis affected individuals. With this technique, the probe or the probe target complex may be labeled in a variety of ways, including the use of radioactive enzymes that produce color change when acting on certain substrates, and fluorescent dyes. This technique may be automated and is currently commercially viable or available in a kit. It's viable and available in a kit form that can detect 12 frequent and 17 rare cystic fibrosis mutations. Another method that has been marketed as a kit is based on the PCR amplification of specific alleles. Using this protocol, several PCR are performed simultaneously for each DNA sample. The primers anneal to different regions for different mutations following amplifications. The presence of a DNA band of a particular size indicates that a specific mutation is present. In this case, different sized DNA fragments are typically separated either by gel electrophoresis or by capillary electrophoresis. This test is quite rapid and it has the ability to detect a variety of different mutations. However, it does not distinguish between homozygotes and heterozygotes, so a positive response must be followed by additional tests to determine whether a positive test is indicative of a cystic fibrosis gene carrier or affected individual. The PCR-only procedure described in detail below is also commonly used to detect cystic fibrosis mutations. This technique is considered to be highly accurate compared to many other protocols and has the highest detection rate of any of the diagnostic tests for this disease. Moreover, it is readily amenable to automation, notwithstanding the success with all of the procedures mentioned above. Researchers continue to refine and develop these and other approaches to the diagnosis of cystic fibrosis. Sickle cell anemia. This is a genetic disease that is the result of a single nucleotide change in the codon or the 6th amino acid of the beta chain of the hemoglobin molecule In individuals homozygous for the defect SYS, the red blood cell, the shape of the red blood cell is irregular, sickle shape because the conformation of the hemoglobin molecule is distorted by a single amino acid change from glutamic acid to valine. The biological ramifications of the genetic alteration are severe anemia and progressive damage to the heart, lungs, brain, joints, and major organ systems. The anemia is caused by the inability of the mutated hemoglobin to carry sufficient oxygen. The life expectancy of SYS homozygotes is quite short. Heterozygous individuals, AIIMS genetic carriers, have normal shaped red blood cells and no symptoms. And unless they are subjected to extreme conditions. such as the high altitudes, or extremes of temperature that are low, with oxygen supply. If both parents are heterozygous, there is a 25% chance that a child of theirs will have sickle cell anemia. That is, will be as biased homozygote. The sickle cell anemia gene occurs with high frequency among Black Africans and their descendants and in Hispanic populations. KORAH screening for the sickle cell anemia is routinely conducted in the United States so that those individuals who are at risk for transmitting the gene to the offspring can be identified. One of the test systems is described below. The single nucleotide change in the beta-globin gene that can cause sickle cell anemia by chance abolishes a CBNL restriction endonuclease site. This restriction enzyme recognizes the sequence CCT-NAGG and claims the DNA between the C and the T. The letter N indicates that any one of the 4 nucleotides can occupy this position in the normal gene. The DNA sequence is CCTGACGG, whereas in the sickle cell anemia gene, the sequence of CCT, GT, G, and G. This difference forms the basis for a DNA diagnostic assay. After oligonucleotide primer sequences that flank the CBNL site are added, a small amount of sample DNA can be amplified by PCR. The amplified DNA is digested with CBNL, and the cleavage products are separated by gel electrophoresis and visualized by ethidium bromide staining of the DNA in the gel. If the CBNL site is present, a specific set of DNA fragments is observed. A different profile of DNA fragments occurs if the CBNL site is absent. By this procedure, the genetic makeup of a tested person can be determined quickly, directly, and easily. Moreover, because of the fortuitous loss of the CBNL site, this assay functions without the need for a target probe hybridization reaction. Let me just repeat that line for you. Moreover, because of the fortuitous loss of the CBNL site, this assay functions without the need for a target probe hybridization reaction. Okay, now we have the PCR by OLA procedure. Obviously, not all genes that produce detective genes affect existing restriction endonuclease sites. Therefore, there are other strategies for detecting single nucleotide changes are required. One of these procedures, we'll come to the diagrams later. Let's finish the subject first. Combines PCR with an oligonucleotide ligation assay. OLA, that is oligonucleotide ligation assay. Let us assume that in a normal gene at a specific site, say nucleotide number 106, the nucleotide pair is AT. In the mutant form, the nucleotide pair at this site is GC. Knowledge of the sequence of nucleotide pair at this site is GC. Knowledge of the sequence of nucleotides on both sides of position 106 enables the design and use of 2 short 20 nucleotide adjacent oligonucleotide sequences that are complementary to one of the 2 native DNA strands. The essential feature of this pair of oligonucleotides is that one of them, ProbX, has as its last base at the 3'. and the nucleotide that is complementary to the nucleotide at position 106 of the normal sequence. The other oligonucleotide, probe Y, starts at its 5' end with a mononucleotide that is complementary to the nucleotide immediately adjacent to the position 106. When these two probes are hybridized with the target DNA containing the normal sequence, which has been amplified by PCR, the nucleotide at the end, at the 3' end of probe X base pairs with the target DNA, and probe Y is aligned so that its 5' end lies next to the 3' end of the probe X. The addition of DNA ligase to the reaction covalently joins probe X and probe Y. By contrast, when these two probes are hybridized to mutant DNA, in which the nucleotide at position 106 is altered, the nucleotide at the 3' end of probe X is mismatched and is not able to pair with the nucleotide 106 and the target DNA sequence. Probe Y, however, is perfectly aligned. In this case, DNA ligase cannot join probe X and probe Y because of the single nucleotide misalignment. Other oligonucleotides probes can also be chemically synthesized to give a perfect base pair match when nucleotide 106 is mutated. Obviously, with the second set of probes, ligation occurs when they are hybridized to target DNA that contains the mutant nucleotide, whereas with normal target DNA, the single nucleotide pair mismatch prevents the ligation of the probes. In short, PCR is designed to distinguish between two possibilities: ligation and no ligation of the two input probes. To determine whether ligation has occurred between two indicator probes, probe X is labeled at its 5' end with biotin, and probe Y is labeled at its 3' end with the deoxygenin, Gen E. Let me spell that again, or pronounce that again, deoxygenin. The low molecular weight compound deoxygenin serves as an antibody binding indicator. After the hybridization, ligation steps are carried out. The DNA is denatured to release the hybridization probes, and the entire mixture is transferred to a small plastic well that has been coated with streptomycin. The well is then washed to remove unbound material, so only the biotin labeled probe DNA remains bound. Next, anti-deoxygenin antibodies, which have been previously coupled in alkaline phosphatases, are added to the well. After an additional washing step to remove unbound conjugated anti-deoxygenin antibodies, a colorless chromogenic substrate is added. The appearance of color in the well indicates that anti-deoxygenin antibodies have been bound to deoxygenin and that the deoxygenin labeled probe was ligated to the biotin labeled probe. If no color appears, then no ligation occurred. With two pairs of probes, it is possible to ascertain the genetic makeup of any tested individual at a particular site. For example, heterozygous individuals yield positive results with both pairs of probes. The DNA from people with two copies of the normal gene gives a positive response only with set of probes that contains the nucleotide complementary to the nucleotide at the normal site. Finally, DNA from individuals with two altered gene copies will give a positive response only with a set of probes that are designed to detect the mutant site. To minimize the amount of the original sample DNA that is required for the assay, the segment of the target DNA sequence that contains a nucleotide site to be tested is amplified by PCR before the hybridization reaction. Overall, the PCR OLA system is rapid, sensitive, and highly specific. It has been automated with a robotic workstation to carry out the steps of the assay procedures. Under these conditions, as many as 1,200 ligation reactions can be conducted per day. The ligase chain reaction assay is a simpler, albeit less sensitive variant of the PCR OLA system. Sample DNA is mixed with excess amount of a pair of OLA indicator probes, as described above, in the presence of a heat-resistant DNA ligase. After an initial ligation reaction at 65 degrees centigrade, the temperature is raised to 94 degrees centigrade to denature the probe target DNA hybrid, and then lowered to 65 degrees centigrade to allow hybridization of the free, non-ligated OLA indicator probes to the target DNA. The cycle is repeated 20 times. If the OLA indicator probes match the target DNA perfectly, then ligation will occur at 65 degrees centigrade during each cycle, and after 20 cycles, enough ligation product probe X joined to probe Y will accumulate to be observed by either jet electrophoresis or an ELISA detection system. If no ligation occurs because of a mismatch, then no joint probe product will be produced or detected. Padlock probes. A padlock probe is an oligonucleotide that is complementary to a target DNA or RNA sequence at its 5' and 3' ends, but not in its middle region. When a padlock probe hybridizes to its target sequence, the 5' and 3' ends of the probe come into close proximity with one another, and the middle portion loops out. Following hybridization, if the probe is exactly complementary to the target sequence, the 5' and 3' ends of the probe can be joined to one another by DNA ligase. The fact that two sequences, actually two ends of the same oligonucleotides, must bind to the target ensures a high specificity of detection for DNA ligation to occur. Both sequences must hybridize perfectly to the target. This makes it possible to easily detect allelic sequence variants. If there is a mismatch at either end, no ligation occurs. Following the ligation reaction, the probe target hybrid can be detected because of promoter molecules, such as biotin, or deoxygenin that are attached to the middle linker portion of the padlock probe. Padlock probes typically have sequences approximately 50 to 29 nucleotides in length and at the 5' and 3' ends that are complementary to the target sequence and a middle region of approximately 50 nucleotides. This procedure has become popular with researchers as it is simpler with fewer steps than the earlier procedure. In addition, the procedure requires one oligonucleotide compared to two for OLA, and it is amenable to automation. And genotyping with fluorescence labeled PCR primers. PCR primers labeled with two different fluorescent dyes can be used in the development of non-radioactive color-based detection systems. To distinguish between mutant and wild-type DNAs, PCR is performed with two different primers. One is exactly complementary to the wild-type DNA and is labeled at the 5' end with the rhodamine red. The other is complementary to the mutant DNA and is labeled at its 5' end with fluorescein green color. In both cases, amplification is programmed by a third unit labeled primer that is complementary to the opposite. We repeat that, a third unlabeled primer that is complementary to the opposite strand. Since PCR amplification can occur only when the primer is exactly complementary to the target DNA, the presence of these primers in the same reaction mixture will result in the amplification of either the wild type or the mutant DNA, or both, depending on which target DNA is initially present to act as a primer as a PCR template. If an individual is homozygous for the wild-type DNA after PCR and removal of unincorporated primer, the reaction mixture will fluoresce red if he or she is homozygous for the mutant DNA. The reaction mixture will fluoresce green, and if he or she has both mutant and wild-type DNA, that is heterozygous, the reaction mixture will fluoresce yellow. This assay can be automated and adapted for any single nucleotide target site of any gene that has been sequenced. The problem with this technique is that it is limited to detecting an SNP. Analysis of multiple loci is not possible, since the presence of many different PCR primers in one reaction tube could lead to large numbers of cross-reactions among primer pairs, with a large number of non-specific PCR products being formed. TaqMan assay. The TaqMan protocol is used to check individuals for the presence of SNPs that are indicative of any of a variety of genetic diseases. Made popular by one particular company, it is based on the 5' nuclease activity of Taq polymerase, which is commonly used to amplify DNA in PCR applications. To simultaneously monitor wild-type and mutant alleles, 2 TaqMan probes are utilized. Each probe is exactly complementary to either the wild-type or the mutant DNA sequence, and each probe has a different fluorescence dye to the 5' end. Intact probes with the bound or unbound cDNA do not fluoresce because of the presence of the quencher molecule at the 3' end of the probe. As PCR proceeds from primer's flanking molecule at the 3' end of the probe, as PCR proceeds from primers flanking the probe hybridization site, the TaqMan probe is displaced by a growing DNA strand, and the 5' nucleus activity of the TaqMan polymerase degrades the 5' end of the TaqMan probe, thereby releasing the fluorescent dye and removing it from the proximity of the quencher molecule. This lecture will be over in a minute and a half from now. I'll be proceeding with the next lecture on the same topic. Please make notes and take necessary screenshots. Once the chapter is over, I'll give you a separate screening of the illustrations and schematic representations, right? Okay. There's only TaqMan probes that were previously bound to target DNA will be degraded and subsequently fluoresce. Any mismatched probes due to mutations in the region where the TaqMan probe binds will be displaced but not cleaved, so they will not fluoresce. By monitoring the fluorescence at 2 different wavelengths, one for each TaqMan probe, it is possible to distinguish the wild-type heterozygotes carrying 1 mutant and 1 wild-type gene, and individuals that are homozygous for the target mutation. In fact, this technique may be used to assay for 2 or more mutations at the same time. The only requirements for the successful employment of the technique are that 1, the precise DNA sequence of the target DNAs must be known, and 2, the fluorescent dyes must have well-separated non-overlapping fluorescence maxima. And onto the summary, which will be completed in the next lecture, along with the review questions. I am just about showing you a couple of illustrations till the subject is over. From the last upward, the first illustration would be, excuse me, for the TACPAN assay. This is how it shows. Please take screenshots and make notes. A TACPAN probe that is complementary to the wild-type DNA is added prior to the PCR. Amplification of the DNA sequence. The probe contains a fluorescent dye attached to its 5' end, green, and a quencher attached to its 3' end, blue. Right? When the probe is intact, the quencher interacts with the fluorophore